Welcome to Ajad Bioacademy. Today I will talk about the basic difference between gram-positive and gram-negative bacteria. In my last lecture, I tried to cover the whole procedure of bacterial cell wall synthesis. Okay, so before going to the details, you should have some basic knowledge about the bacterial cell wall synthesis. Today I will actually try to cover in molecular level. That means in molecular differences between gram-positive and negative bacteria, which molecules are very specific in gram-positive and which molecules are very specific in gram-negative bacteria, and what are their basic roles. And actually, I also try to cover why the gram-negative bacteria are very difficult to kill than the gram-positive bacteria. And also, I will try to cover the basic antigenic molecules among the gram-positive and gram-negative bacteria. Okay, so now one after another, I will try to explain in molecular level. That means, firstly, I will say this is your gram-positive bacteria, and this is gram-negative bacteria, and this is the component, actually component, and this is gram-positive, and this is gram-negative. Okay, so first one I can say that is plasma membrane. Plasma membrane. So I will not take any more time on the plasma membrane. Actually, you know, the plasma membrane is the basic inner membrane of a cell, which will just carry the interior components of the cell. Okay, so this is very common and very essential for each and every cell. Okay, so as gram-positive and gram-negative, both are cell, bacteria both are cellular organism, so it has some external structural difference, but inner membrane, that is plasma membrane, is common. So I will say in both gram-positive and gram-negative bacteria, plasma membrane is positive. Okay, just simply you can say something is needed to retain that will act as a primary outer membrane. Okay, so that's it. So second one is, I will go for periplasmic space. Actually, periplasmic space means here it is very easy to understand if I just go first in gram-negative bacteria, because here it is clearly mentioned here that this is the inner membrane, that is plasma membrane, and this is the outer membrane. Okay, so inner membrane and outer membrane, and in between a clear space is there. So this is the space in between inner and outer membrane. So inner membrane is the plasma membrane and the outer membrane, and in between the space is periplasmic space. Okay, so periplasm means the space between inner and outer membrane. Okay, in a sense you can say the space of inner membrane and other layer if it is present in a cellular component. Okay, so now come to the gram-positive. Is there any outer membrane here? No. Here is the outer membrane. This is the outer membrane, and this is the inner membrane. But here, no outer membrane. So if there is no outer membrane, so how I will define the space between inner and outer? And if I consider this is the inner membrane, that is plasma membrane, and this is the another layer, that is the peptidoglycan layer, which will I just discuss now, just after this point. So where is the space? It is very difficult to define. And if I just try to figure out that this space is the periplasmic space, because this is in between the inner membrane and the outer layer. So you can say this is the periplasmic space. So it is very difficult to define. Okay. So what I wanted to say, that is, in the gram-negative bacteria, periplasmic space is present. I just noted it by simple positive symbol. Okay. Or you can say, no, this is not periplasmic space is present, as it is very thin and it is not possible to define. That means it is not well defined. Not well defined. Okay. You can say positive but not well defined, or also you can say negative. Okay. But here the periplasmic space is very thick and it is clearly visible or clearly notable. So periplasmic space is present and well defined, and you can say very thick. Okay. So this is periplasmic space of gram-positive and negative bacteria. Number 3, I will say, is the peptidoglycan. This is very much important molecule of gram-positive and negative bacteria. In a sense, you can say, based on this peptidoglycan molecule, gram-positive and negative just differentiated in the earlier stage. Okay. So now, what is peptidoglycan? So if I just try to show you, this is the peptidoglycan. Here, this layer, this peptidoglycan, and this portion is peptidoglycan. So if I just try to give you some details, that is, here is the peptide, that means peptide, that means amino acid chain, and also glycan, that means sugar. So this layer, peptidoglycan, is made up of sugar and peptide, amino acid chain. So where is that? So now, this blue color is noted here as NAG, that is N-acetyl glucosamine, and very adjacent to the NAG, in green color, it is noted that NAM, that is N-acetyl muramic acid, and one NAG is attached with the NAM, that is N-acetyl muramic acid, by the glycosidic bond. You know, already I discussed in my previous lecture. Okay, so I will not go for details. And you know, the NAM has some capacity to carry the pentapeptide. This is pentapeptide. Okay, so in that way, NAM NAG, and here another NAM NAG, and then NAM NAG, it will go on. So this is the sugar skeleton chain, and this is the peptide, pentapeptide. So peptidoglycan, peptide and glycan. So peptidoglycan. So peptidoglycan monomer, that is peptidoglycan, that is NAM-NAG, and pentapeptide is a basic unit, and it will attach to each other again and again. The sugar chain will be attached by the glycosidic bond, and the peptide and another chain of NAM peptide will be connected by the pentaglycine, or you can say directly connected by the covalent bond. So if I just try to show you, here is the pentapeptide, which is attached with the NAM. You know the NAM has capacity to carry the pentapeptide. So this is the pentapeptide. And here this NAM is also carrying the pentapeptide. And in between the pentapeptide, here is a bond, cross link. Here is the cross link. Here is the cross link. And the cross link can be made up of pentaglycine. You can say in Streptococcus, pentaglycine cross-link is available. If you think about the E. coli, then you can say directly covalent bond, or you can say amide or peptide covalent bond. Covalent bond. That means direct bonding. Okay, this cross-linking. So now come to the point, because my not focusing point is to again explain the peptidoglycan. Main focusing point is the difference. So what is the difference? So this, the sugar chain, another sugar chain, another sugar chain, and two sugar chain is attached with the pentapeptide cross-linking, and then the peptidoglycan is the layer here is multiple layer. Okay, so I will just now go for the difference, that is peptidoglycan. Of course, here it is present and it is multi-layer. It is multi-layer that can be up to 50 layers. So single layer, double layer, triple layer, multiple layer, and it is very thick. Very thick, and the thickness could be 20 to 80 nanometer. That means very highly thick. So what is the basic things of peptidoglycan based on the gram-positive bacteria is that the gram-positive bacteria outer cell membrane, it will carry just multiple layer of peptidoglycan, which is basically can be up to 50 layers, and the total thickness can be 20 to 80 nanometer. That is very thick. Okay, but compared to gram-negative bacteria, if you look here, the layer is single or double. That means in gram-negative bacteria, the peptidoglycan just only here it is present, but peptidoglycan layer is very thin, as it is single or double layer. Okay, so it is very possible single or double layer, so of course it would be thin, and the layer thickness is only 2 to 3 nanometer. Okay, so if you just now look, this is the gram-negative bacteria. This is the peptidoglycan layer, and don't get confusion with the periplasmic space. That is the space where peptidoglycan is there. Okay, and peptidoglycan is the different things which is made up of peptide and sugar. Okay, so it's very single or double layer, and this is the here. If you have a look here, so this is the multiple layers. Okay, so the basic difference of peptidoglycan, the gram-positive bacteria, is very thick and very thin. And now I'm just coming to the fourth point, that is, I will say, outer membrane. Outer membrane. So outer membrane, you know, already I said when I just tried to say something about a periplasmic space, then I said outer membrane and inner membrane. So here, outer membrane is clearly visible, but here no outer membrane. That means here no outer membrane, and here outer membrane is present. Okay. Now, As outer membrane is present, so here you can see the outer membrane is just carrying some molecules, that is lipopolysaccharide, which is directly integrated with the outer membrane. You can think about the porine, which is a channel which is also present in the outer membrane. If you think here some embedded protein, here some embedded protein also present in outer membrane. So those things will be very possible to present in gram-negative bacteria, but it's not possible to present in gram-negative bacteria, as gram-positive bacteria has no outer membrane. So as gram-negative bacteria has outer membrane, so outer membrane containing or carrying molecule will be present in gram-negative bacteria, and that not be possible in gram-positive bacteria. That's it. So I will just now try to say something about those components in details. So fifth point is lipopolysaccharide. Okay, so already I said lipopolysaccharide is only present in gram-negative bacteria, but here lipopolysaccharide is absent. So what is the basic function of lipopolysaccharide? I will explain when I will try to cover the antigenic properties. Then I will give you details. But now I will say lipopolysaccharide is basically made up of lipid, saccharide, that is oligosaccharide, and O antigen. There are three parts of this lipopolysaccharide. And that lipopolysaccharide is basically the integrated part of the bacterial cell wall of the gram-negative bacteria, which basically integrated within the outer membrane, and which will make the structural stability of the bacterial cell wall of gram-negative bacteria. Okay, as it is incorporated into the cell membrane, and also it has antigenic properties. Okay, and a part of this lipopolysaccharide is considered as a endotoxin. Okay, so next one is porine. So porine is also the major component of outer membrane of gram-negative bacteria. So here porine, of course, in gram-positive bacteria, porine will be absent, and here porine will be present. So what is the function of porine? So you know the porine is basically it's just like a channel, okay, made up of protein that will maintain which the favorable things or substances will be allowed to enter into the cell or not. That means it will regulate or control only suitable substances can get chance to enter into the inside, other than it will not be allowed. Okay, and also the bacteria, gram-negative bacteria, will efflux from unwanted substances to the porine. That means gram-negative bacteria, the porine is important part of that outer membrane. Okay, but here no outer membrane, no porine. Okay, so now I will go for another things is that is the another part is now 7 embedded protein. Actually, this is a very common embedded protein. Here, if you just look, this is embedded protein, and this is embedded protein. So embedded protein are present in both gram-positive and gram-negative bacteria, but actually embedded protein are different in gram-positive and gram-negative bacteria. So here, this is embedded protein is present embedded protein is present. But here embedded protein can be, M protein, you can say protein A, like that. And here embedded protein, you can say outer membrane protein A, you can say this is the embedded protein. And here also coding and also embedded protein. Coding has some part like outer membrane protein C, outer membrane protein F, like that. Okay, so these are the embedded protein, that is completely different from gram-positive and negative. So now I will go for the specific component of gram-positive bacteria, that is teichoic acid and lipoteichoic acid. This is very important because gram-negative bacteria has outer membrane, and outer membrane has the lipopolysaccharide, which is incorporated into the membrane and which is basically just act like a integral part of bacterial cell wall. That makes a very stable bacterial cell wall. Okay, so who will make it stable? Because he has the multiple layer of peptide glycan. So by two sugar chain is attached by the peptide containing cross-linking, but overall the peptide glycan, how it will be stable. So here is also a support molecule is important. So that molecule is teichoic acid and lipoteichoic acid. Teichoic acid basically incorporated into the peptide glycan. That means it is attached within the peptide glycan and some proceedings in outside. And here the lipoteichoic acid. What does it mean? Actually, lipo, that is lipid part, that means that it is connected. That's lipoteichoic acid is completely going through into the membrane part, that is plasma membrane. And you know the plasma membrane is made up of lipid bilayer. So in the lipidic part, it is incorporated and it is just protrude outside of the peptide glycan. So it is connected to the lipid, so lipoteichoic acid, and this is the lipoteichoic acid. Both are very integral part of the bacterial cell wall of the gram-positive bacteria, which makes the peptide glycan very stable, very rigid. Okay, so this is the teichoic acid and lipoteichoic acid. As peptide glycan layer is very thin, that is single or double layer, so it is necessary to make them stable or make them make any supporting molecule is needed in a common sense. No. So teichoic acid and lipoteichoic acid is not present in gram-negative bacteria, as it is concerned with the peptide glycan. So peptide glycan is very thin, so it is not important, not necessary. So here teichoic and lipoteichoic acid is present in gram-positive bacteria, but absent in gram-negative bacteria. Okay, so this is all about the gram-positive and gram-negative bacterial basic difference. So among them, which substances are antigenic and what other substances can be antigenic also? So I will just add another point, that is antigenic component. Antigenic molecular component. Antigenic component, I will say, here I will just try to explain here the antigenic substances. So antigen means, what does it mean? Antigen means the substances, basically protein in nature, when the foreign substances basically foreign microorganism will enter in your body. The microorganism, the whole microorganism can act as an antigen. That means if you try to catch something, you will catch the hands or catch your shoulder or catch your throat or catch your hair. But is it possible to capture the whole body at a time? Not possible. So your defense system, your defensive cell that basically your WBC, different cellular WBC, someone is responsible for engulfing, someone is responsible for recognition. So actually, the which substances will trigger to produce some weapon, and that weapon will interact with that substances. The weapon name is antibody, and the substances on which it will interact, that is antigen. So antigen and antibody, highly specific in their action, and antigen and antibody are always enemy to each other. Always, never be friend to each other. So same antigen and antibody always be absent. That means it's not possible to just to live together, not possible. Okay, so antigen and antibody. So which substances will just manage to produce antibody? Those substances are called antigen. That's it. So what type of substances from gram-positive bacteria and what substances of gram-negative bacteria will act as an antigen? So now I will cover here now. So if I just go with the gram-positive bacteria, so now I will say, I will just mark by green color. This is the antigen, teichoic acid, lipoteichoic acid. These are highly antigenic substances. Okay, some embedded protein. I will just mark it by green color. So embedded protein name is M protein. These are the antigen, and I will say protein A for gram-positive bacteria. M protein, very available in streptococcal bacteria. So the teichoic acid, lipoteichoic acid, M protein, protein A, these are the highly antigenic. And also the peptidoglycan. The peptidoglycan, the whole peptidoglycan is, it will act as antigen. It's very complex matter, but the part of peptidoglycan will act as an antigen. That means, in a sense, you can say the peptidoglycan can act as an antigen. Okay, so this is also an antigen. So peptidoglycan, a part of peptidoglycan, the fragment of peptidoglycan can act as an antigen. And also, the gram-positive bacteria will create some substances, like these substances, and it will exit from inside to outside, and it will act as antigen. Suppose these are the antigen, and these are called exotoxin. Exotoxin. So for gram-positive bacteria, teichoic acid, lipoteichoic acid, embedded protein, that is M protein, protein A, and exotoxin, peptidoglycan can act as an antigen. Okay, that can trigger to just produce antibody from your immunocell, basically beta cell, that is plasma cell. Okay, now come to the gram-negative bacteria. Gram-negative bacteria, the antigenic substances, I will mark it in a red color. That is, this is lipopolysaccharide. And already I said, lipopolysaccharide is an integral part of outer membrane of the bacterial negative bacteria, and lipopolysaccharide is lipid, saccharide, and O antigen. So this is highly antigenic. Okay, lipopolysaccharide. And the part of lipopolysaccharide is called endotoxin. Endotoxin. It is just come out from inside to outside, so exotoxin, and this is the part of outer membrane lipopolysaccharide, that is the endotoxin. So this is endotoxin, can act as an antigen, and lipopolysaccharide itself an antigen. Purine. Purine also can act as an antigen. Okay, and here some embedded protein. Already I discussed, that is OMPA, that is outer membrane protein A, outer membrane protein C, F. These are the embedded protein, can also act as an antigen. So these are the antigenic component of gram-positive and gram-negative bacteria. Okay, and also here, although it is very thick, but can act as an antigen, that is the peptidoglycan. So this is the peptidoglycan antigenic substance. So this is all about the gram-positive and gram-negative bacterial basic molecular difference. Okay. Before just going to end, I will just ask you why it is gram-positive and gram-negative bacteria. Actually, I didn't cover it. Okay, so now I will just shortly explain why it is gram-positive and gram-negative, and just I will briefly explain the gram staining technique, very briefly. Okay, only regarding the gram-positive and negative. Actually, not the whole procedure of gram staining technique. So now come to the why it is called gram-positive and why it is called gram-negative. Actually, Hans Christian Gram was the discoverer of gram staining technique, and based on the staining technique, this is gram-positive and gram-negative. Okay, so this is the gram. And staining. So how does it mean? Stain. Stain means color. Okay. So by putting different types of color, we will identify this is gram-positive or this is negative. So very briefly, I will just try to explain. Okay. So now I will incorporate one color. One color I will add here. So think this color is crystal violet. Okay. When I will put crystal violet here, so it will just go into the peptidoglycan layer very easily, very quickly, as it has no any outer membrane. So peptidoglycan, within the peptidoglycan, crystal violet will incorporate very easily. If I just put crystal violet in gram-negative bacteria, it has outer membrane. So firstly, it will be it will not incorporate into the peptidoglycan layer very easily because outer membrane. And also, if it is it can pass through the outer membrane, then very little amount it can enter into the peptidoglycan. And also here, peptidoglycan layer is very thin. So it will be captured by the peptidoglycan chance is very little. Okay. So next, we will just add here another things, that is iodine. Okay. So when iodine will be incorporated, then iodine will bind with the iodine easily go into the peptidoglycan and it will bind with the crystal violet. Okay, so crystal violet and iodine will make a complex. And where the complex formation will happen? It will happen within the peptidoglycan of the gram-positive bacteria. Okay, so now here, if you just add iodine, the iodine incorporation of the iodine chance is very less by the outer membrane, and the binding with the peptidoglycan, binding with the crystal violet is also less, as the peptidoglycan layer is very thin and outer membrane protection is there. Okay, so now we will just wash out by alcohol. You just put alcohol for washing purpose. Actually, I said I will not go details because I will take a slide and I will just put bacteria and it will dry and the heat struck, and then I will wash water, put crystal violet, and it will keep some time to dry, and then put water by the extra staining wash out. I'm not going to the details actually. Okay, so now the iodine has binding chance is less, so on both bacteria, alcohol is added for washing. Then what will happen? Alcohol will interact firstly with the outer membrane because this is the lipid. So it will mostly degraded by the alcohol, and also it will wash out the little amount of crystal violet and iodine. It will wash out by the alcohol. But here it is already incorporated into the multiple layer of the peptidoglycan. And when alcohol is added, no chance to wash out because already make a big complex with crystal violet and iodine within the net of the peptidoglycan. And here no chance. So what will happen? The crystal violet color, or you can say purple color will be here, but here is no color. So no color means negative, and color is here, so positive. So this is gram positive, this is gram negative. Okay, so this is all about. But you can just counter stain. Another color you can apply. After wash out, you can apply safranin. Then what will happen? Safranin color, if you add here and if you add here, then what will happen? Safranin color has itself color, like brick color, that is red, or you can say pink. So when it will add here, then if you just wash by water, then it will just watch under microscope, then the safranin color here, pink or red color will be visible here. Pink or red color. And when safranin will be added here, then no color change. Why? Safranin has itself as a color, but you are applying here, but color is not changed because already that peptidoglycan layer is already blocked by the crystal violet iodine complex. And crystal violet has a color and very prominent and already saturated. But when you are just applying safranin, the safranin color is not able to beat the color of the crystal violet iodine. Okay, so it's just keeps its original color, the first color. And this is not possible here because only it is visible by safranin color. Simply you can say this is very strong red, and on this strong red if you apply some red color, so is it possible to beat this color? No. So just like that. So this is the gram positive and gram negative staining and the basic difference. Okay. So before going to add, actually previously I said I will cover why the killing of gram positive bacteria is easy than gram negative. Okay, that means why gram negative bacteria is difficult to kill. And that's why I will just shortly explain now. Just very simply, you can think, why? If you apply a weapon, that means chemical, just if you apply antibiotics here and here, what will happen? It will incorporate very easily within the bacterial cell membrane and then inside, because it has no any outer membrane. But here, this is the outer membrane. and outer membrane is highly regulated and controlled by the lipopolysaccharide porine, basically porine, because the outer membrane is as a protective layer and also regulating which favorable things will enter through the membrane or not, through the porine or not. If unwanted source has entered, then easily it can reflux through the porine, reflux through the membrane. Okay, so the two things here. One is easily antibiotic cannot enter into the gram-negative bacteria because of the outer membrane. Second thing is that if it is entered, then it is also has higher chance to exit from the inside to outside by flux, by the porine channel. But here it is very easy to enter into the cell, and it can interact with the peptidic glycan, that is cell wall. It can interact with the membrane, and it can also interact with within the internal genetic material or ribosome or like that. But here it is actually difficult to enter. This is the basic difference. So if you just ask, if I just ask you which one is very difficult to kill, so of course gram-negative bacteria is very difficult to kill because of their outer membrane, because of their porine, that is efflux character. And another things you can add here, that is the gram-negative bacterial cell wall cell division rate is high, because here the peptidic glycan layer is very thick. When cell division will occur, so all the components, cellular component is important. So when bacterial cell wall will be developed, then it will be separate individual bacteria. Okay, so the gram-positive bacteria cell division is rate is slow and is high. That means it will quickly cultivate and quickly multiply, and it's less. So it's very difficult to kill by antibiotic, and also its production rate is high. So overall, the gram-negative bacteria is very difficult to kill. Okay. Although some basic difference of gram-positive and negative bacteria, some are intracellular, some on gram-positive bacteria, some external different character. So that is the different things. But usually, in generally, gram-positive bacteria is very difficult to kill than gram-positive bacteria. That's all about today's lecture, gram-positive and gram-negative bacteria. So thank you very much.